Good afternoon, everyone. My name is Anita Ramani, and I'm here to talk to you about glycolysis, gluconeogenesis, and the role of aldolase gene expression. Today, we will be describing the purpose of glucose, explore how glycolysis produces energy, illustrate how gluconeogenesis produces energy, and identify how transcription factors regulate expression of aldolase. We are going to begin with glucose. It is also known as sugar or blood sugar. Glucose is the simplest monosaccharide. This is the form that it can be in its simplest. Glucose is a hexose sugar, meaning it is a monosaccharide with 6 carbon atoms and 6 oxygen atoms. You can see this shape it's in is in a hexose configuration. Carbohydrates are the universal energy source for every organism. They are essential in fueling aerobic, which means with oxygen, and anaerobic, without oxygen, cellular respiration. Cellular respiration is a pathway that breaks down glucose and produces ATP that our bodies need to function. Carbohydrates typically enter as a variety of sugars known as galactose and fructose, lactose, sucrose, or even starch or fiber. Then it is broken down through processes into glucose. In the body, excess glucose is stored as glycogen, a glucose polymer, which are stored as long chains of glucose. These polymers are utilized during fasting. Glucose is absorbed and transported directly to the body cells to fuel their metabolism. Several tissues require continuous glucose supply, including the brain, erythrocytes, our renal function, the lens and the cornea, the testes and the ovaries, and even our skeletal muscles during exercise. So through these processes, we also produce byproducts, carbon dioxide and water, as shown here. So we're going to jump right in. Where does this all start? This is an image of the process of cellular respiration. We are going to start with glycolysis, which occurs in the cytosol of the cell. The cytosol is within the cell, and it is a jelly-like substance that surrounds the organelles. As you can see here, there is the mitochondria. This is an organelle within the cell. The process of glycolysis begins in the cytosol area, as you can see here. Glycolysis is the breaking down of sugar or glucose molecules to release energy that is needed for furthering cellular respiration. As you can see here, through glycolysis, some ATP is produced. ATP is also produced here in the citric acid cycle, which we will dive into deeper in a little bit. And then the electron transport system also creates ATP. ATP, again, is our primary energy source for important biological functions. We need it for our everyday activities. Glycolysis is the first step of cellular respiration that produces 2 molecules of pyruvate after the breakdown of a glucose molecule through a set of enzymatic reactions. So you see here, glucose molecule is right here. Through a series of enzymatic reactions, you end up with 2 molecules of pyruvate. Glycolysis can occur in an aerobic or anaerobic situation. Again, it does not require oxygen. Each glucose can be broken down into 2 pyruvate molecules. In this phase, 2 ATP are used, as you can see here, and then once the phosphate is removed, it is now 2-ADP. So in this phase, 2-ATP is used to produce 4-ATP, but the net gain is 2-ATP because 2-ATP were used to allow this reaction to occur. So once pyruvate is created, there are two options. So for example, if there's intense exercise, pyruvate will convert to lactate through anaerobic glycolysis. So if there's not enough oxygen, it will produce lactic acid. And aerobic respiration, as we talked about, the net production of ATP is 2 molecules. So the cytoplasm of the cell can produce lactic acid when both glucose and pyruvate are present. In this situation, that glucose converts to pyruvate, which is then converted to lactic acid to produce energy for your cells. But again, it's anaerobic, so it's without oxygen and it's less efficient. But even in this situation where lactic acid is produced, it cannot enter the mitochondria to produce more energy through the citric acid cycle. So aerobic is the most efficient because then with the oxygen, pyruvate can enter the Krebs cycle or the citric acid cycle. And in the end, as pyruvate undergoes oxidative phosphorylation or processes leading to the production of ATP, 38 ATP are produced. Sometimes it can get confusing to understand the production of ATP and how it occurs and the numbers of ATP that are produced. So we're still covering glycolysis, and as you can see here, the glycolysis, as we've talked about before, will eventually split into 2-pyruvate. In the cells that use aerobic respiration as the primary source, the pyruvate formed from this pathway will go into the Krebs cycle. So the first phase, you can look at it as more of an investment phase. So this 2-ATP, they're used to begin the process. And then the second is the payoff phase. So 2-ATP are produced. That is what is gained. So in this phase, 4-ATP are produced, but 2 were used, so the net gain is 2-ATP. You can also see here 2-NAD, which becomes 2-NADH. We're going to talk more about what this is and the importance of this in the next slide. So you see NAD plus here. It's a reversible reaction to NADH. So NAD is an electron carrier because of its ability to carry electrons from one region of a cell to another. The structure of NAD provides a safe shuttle for the movement of electrons across the internal regions of the cell. Without molecules like NAD, electrons would not be able to move to its proper destination. This process of moving electrons makes NAD and NADH an oxidation, reduction, or redox molecule. So what is that exactly? So oxidation and reduction are terms we use to describe when we either gain or lose electrons. Reduction is when a molecule gains electrons, such as when NAD plus gains the electrons to become NADH. For example, you can say that NAD has been reduced to NADH. Oxidation is when the molecule loses its electrons, such as when NADH loses its electrons to become NAD. For example, you can say NADH is oxidized to NAD. It is not a permanent situation. So NAD and NADH, again, it's a reversible reaction. It's a reversible cycle. It is not permanent, and it can occur over and over to the same molecule. So in aerobic glycolysis, NADH is transported to the mitochondria where it is reoxidized, lose electrons to NAD, while it participates in the electron transport chain to produce ATP. We are going to dive a bit deeper into glycolysis so you can see some of the enzymes involved in this reaction. First, I want to start with the term sequential mechanisms. This is when a molecule needs to bind to an enzyme so that a chemical reaction can take place. For example, we're going to start with hexokinase up here. It is the first enzyme involved in glycolysis. In this reaction, phosphate is added to glucose. So where does the phosphate come from? It comes from ATP, as you can see here. ATP is converted to ADP, and that phosphate is taken. Then phosphohexose isomerase rearranges the atoms in which has become glucose 6-phosphate, and this process is called isomerization. Jumping down a little bit, aldolase and phosphofructose kinase 1, or PFK1, are also enzymes that have important roles in glycolysis. PFK1 will cleave or split the product of the PFK reaction into fructose 1,6 biphosphate, into glycerolaldehyde 3-phosphate, and dihydroxyacetone phosphate. So aldolase is a sequential enzyme to PFK in glycolysis. Aldolase and PFK1 are the two that regulate the speed of glycolysis. Both must bind to the enzyme before any products are made or released. Again, another example of sequential mechanism. Glycolysis begins with a molecule of glucose and concludes with 2 molecules of pyruvate or pyruvic acid. Before jumping into the next topic, I just wanted to summarize glycolysis. So here in this slide that I showed earlier, glycolysis occurs in the cytosol, and then the pyruvate can enter the citric acid cycle if there is oxygen present. And as you can see here, the NADH is carrying its electrons, and so it travels from the cytosol outside and travels into the mitochondria. The next topic we are going to talk about is gluconeogenesis. So the literal meaning of gluconeogenesis is glucose new formation. So basically, it is synthesizing glucose molecules from precursor molecules, such as pyruvate, which we know all about, lactate, glycerol, which is part of the fat molecule, or amino acids, which are the building blocks of protein. Gluconeogenesis mostly occurs in the liver and the skeletal muscle. It occurs due to a change in homeostasis. For example, if the blood sugar levels drop below a certain level, the liver increases the rate of gluconeogenesis to carry out ATP synthesis, which, as we know, is used for cellular functions. However, if blood sugar rises above homeostatic levels, the liver decreases or slows down the rate of gluconeogenesis to give the body time to store it. Another example is during intense activities such as exercise, running, jumping, etc. If glucose stores in the skeletal muscles drop during times of intense activity, the skeletal muscle increases the rate of gluconeogenesis to create energy. So gluconeogenesis primarily occurs in the cytoplasm or the mitochondria. Gluconeogenesis mainly occurs in the liver. However, during prolonged starvation, kidneys become a major glucose-producing organ. So one of the first steps is to make oxaloacetate. So you would need pyruvate, and the enzyme pyruvate carboxylase will convert pyruvate to oxaloacetate. And as you can see here, 1 ATP will be utilized. NADH will convert oxaloacetate to malate so it can be transported out of the mitochondria. Then it will be oxidized back to oxaloacetate, and it can go through the gluconeogenesis pathway. Gluconeogenesis plays a crucial role in homeostasis. During fasting, when glycogen stores become depleted, our body relies on an internal glucose production source from within. Gluconeogenesis is much like glycolysis, only the process occurs in reverse. As we learned, glycolysis leads to the production of pyruvate, as seen right here, the purple line, from glucose to pyruvate. From here, 3 additional enzymes are required for the conversion to return to glucose. So glycolysis is a catabolic reaction. It is a breaking down process of glucose. And gluconeogenesis is an anabolic, a building up process. This process requires 3 additional enzymes to make up for the 3 irreversible enzymes of glycolysis. Hexokinase is replaced with glucose 6-phosphatase, as you see right there. PFK1 is replaced with fructose 1,6-biphosphatase. And pyruvate kinase is replaced by PEP carboxykinase 1. PEP stands for phosphophenol pyruvate. Glycolysis is the breakdown of glucose, and gluconeogenesis is the formation of new glucose. Glycolysis takes place when cell energy is low and ATP or energy is needed. Gluconeogenesis is the formation of glucose from other sources, such as proteins and fats, and gluconeogenesis occurs when cell energy needs are high, for example, starvation. When you first see these diagrams, you wonder if glycolysis and gluconeogenesis is perhaps a reversible reaction, but it is not. If you look at the outer arrows here, there are 3 reactions on either side that are irreversible. As we talked about in the previous slide, one of the irreversible reactions is pyruvate to oxaloacetate. This enzyme, pyruvate carboxylase, is one of the enzymes needed to catalyze this reaction. Then if we look further up here, the removal of a phosphate from fructose 1,6-biphosphate by FBP1 And then the third irreversible reaction is the dephosphorylation, or the removal of a phosphate from glucose 6-phosphate. So those are the three irreversible reactions. To point out a reversible reaction, here is one right here, aldolase. And as you can see, the arrows go up and down. And so it shows that fructose 1,6-biphosphate with the enzyme aldolase converts to glycerolaldehyde 3-phosphate. Aldolase, or aldolase A, catalyzes the reaction shown here, where two 3-carbon compounds are joined to produce a 6-carbon fructose molecule. Aldolase A is also known as fructose biphosphate aldolase. If a cell is producing glucose, then there will be a steady supply of substrates, which means that there is a substance upon which an enzyme can act upon that flows into the reaction. Once no further fructose 1,6-biphosphate is needed, it will be broken down back into 2-carbon molecules at the same speed. So this is what makes this reaction reversible. It's able to be broken down or created with the help of aldolase. Also, when ATP levels are high, it also inhibits the production of fructose 1,6-biphosphate. And when ADP levels are high, then activation will occur and more fructose 1,6-biphosphate will be created. This allows the cells a way to regulate the breakdown or creation of glucose. As we have discussed, aldolase is a protein-based enzyme that assists breakdowns of sugars such as glucose and fructose to produce energy. It is specifically expressed in the hippocampus of the brain. There are 3 aldolase isoenzymes, A, B, and C, and are encoded by 3 different genes that are differentially expressed during development. Here I'm going to talk about isoforms. Isoforms are different molecular forms of an enzyme. So as we've talked about, aldolases play an important role in glycolysis and gluconeogenesis, and they are present in 3 different isoforms: aldolase A, B, and C. They're also known as aldo A, aldo B, and aldo C. Aldolases are differentially expressed in human tissues, and abnormal expression has been seen in human diseases and cancers. Aldolase A is primarily seen in muscle and red blood cells. Aldolase B is seen in the liver, kidney, and small intestinal areas. And aldolase C is seen primarily in the brain and neural tissue. The expression of aldolase can be regulated by transcription factors such as HNF1, HNF3, CEPB, and DBP. These proteins affect the binding of RNA polymerase to a particular gene on the DNA molecule, and consequently affects the rate of transcription. These factors can cause changes in gene expression. They can either promote or inhibit RNA polymerase by causing a change in transcription. Dysregulation of these transcription factors have been linked to a variety of diseases. For instance, dysregulation of HNF1 or HNF3 has been connected with diabetes and liver diseases. Therefore, understanding how these transcription factors regulate gene expression can help in the development of therapeutic strategies for these conditions. To provide an example, the HNF1A gene provides instructions for making a protein called the hepatocyte nuclear factor 1 alpha, which we know as HNF1 alpha, as a transcription factor, which means again that it attaches or binds to specific regions of DNA and helps control the activity of certain genes. You can see here the multiple roles that HNF1A has. It includes lipid metabolism, so again the liver, insulin, which includes the pancreas, glucose metabolism, which we talked about, glycolysis and gluconeogenesis, and then water reabsorption in the intestines. I would like to share a study involving aldolase. Creatine kinase is a muscle enzyme involved in energy supply to the muscles, while aldolase, as we know, is expressed in multiple organs, and now I will also add the brain as well as red blood cells. So the elevation of serum CK and aldolase they're both considered biomarkers for muscle damage. Usually, high CK levels is often accompanied by high aldolase levels. So the amount of CK elevation is typically correlated with the number of necrotic fibers. So necrotic fibers is basically muscle fiber death. This leads to irreversible damage to muscle fibers. In skeletal muscle, aldolase is abundantly expressed in myoblasts. Myoblasts are cells that can develop into muscle tissue. In these cells, CK expression is typically very low. The question is, even though the patients may have normal CK levels, is the immune-mediated destruction of these myofibers associated with the aldolase increase? In this particular study, 34 patients with various types of myopathies were identified, and myopathies were actually treatable in 27 of the 34 patients. High levels of aldolase were found in all 19 tested for dermatomyositis. And in these patients, they had higher creatine kinase levels. With patients that had higher aldolase and CK levels, they had more frequent symptoms of muscle fiber changes and more rapid irregular contraction of muscle fibers. So in this condition, it usually starts with a skin rash that usually occurs before or with the episode of progressive muscle weaknesses. Elevated levels of aldolase were seen in more myopathies than originally expected, and several of these were treatable. So by testing for both aldolase and CK, it increases myopathy detection compared to just testing CK alone. So it is recommended to check levels of both with those patients that are suspected with myopathy. So this emphasizes the importance of measuring aldolase. Thank you, and are there any questions? Here are my references. Thank you.